Welcome to the Deep Dive. We are here to turn overwhelming source material into those crisp, actionable insights you need. Today, we're diving into something huge. Drug discovery. It's famously slow, right? It takes maybe a decade, costs billions for just one success.
Absolutely. And it's always been the playground of, well, giant corporations, big pharma.
But what if that changes? What if someone challenges that? Not with another huge institution, but, one person using tools available to almost anyone.
That's exactly the story here. We're looking at a system designed to show that, big breakthroughs don't always need a billion-dollar lab.
Yeah, the source material centers on Mansour Ansari, right? A retired software engineer.
That's him, running QuantumLaso, LLC, apparently from like a cluttered back office in Oklahoma, which I think is fantastic.
It really is. It perfectly captures the mission, proving you don't need massive overhead for serious innovation. He's basically meshed classical chemistry with the unpredictable element of quantum entropy.
And the roadmap he's laid out is incredibly practical. We're going to unpack the pieces. Open source software, things like RDKit, Vina.
Stuff anyone can get.
Exactly.
Yeah.
Plus massive public data troves, PubChem, ChEMBL. And then there's AI for the heavy lifting on filtering.
And the quantum piece, which sounds expensive, but maybe not.
Right. Using cloud access to quantum hardware like D-Wave and IonQ, it's about smart access.
So our mission for you today is to map this out, show you the blueprint, really, prove that this frontier of discovery, it's maybe closer than you think.
Like Ansari himself says, sometimes you just have to be the person who will not wait for permission to innovate.
I love that. Okay, let's start where any discovery system has to.
Yeah.
The classical foundation, the tools everyone uses.
Right. To really get what's new here, we need a little context. We went from, chewing willow bark ages ago to the industrial age, synthesizing stuff in labs.
Big vats of chemicals.
Pretty much. Then the digital age hit in the 80s, 90s, computational chemistry, molecular docking. Suddenly, the computer was a virtual lab bench.
Simulating how drugs might work before making them.
Exactly. That's the scaffolding. Even this new system builds on that.
Okay, so let's unpack those classical tools, the essentials. First up, the raw materials, the data.
Yeah, data's key. 3 big players here. First, PubMed. Think of it as the knowledge base. Over 38 million biomedical papers. You use it to understand the disease, the target pathway.
Got it. Knowledge. Then what?
Then ChEMBL. That's the bioactivity database. Crucial stuff. It connects molecules to actual results, what happened in experiments, how well things bind.
So linking the chemical to the biological effect.
Precisely. And finally, the chemical universe itself. PubChem. Over 118 million compounds listed. That's your library of potential drugs to screen against your target.
Okay, data layer covered. What about the software, the workbench?
You start with RDKit. It's often called the Swiss Army knife for this kind of chemistry informatics.
What does it do, though?
Well, it handles chemical structures, sure. But more importantly, it calculates properties. Things like log P.
Log P gets mentioned a lot. Why does it matter so much?
Log P basically predicts if a drug can get absorbed. Its solubility, its permeability, can it actually reach the target in the body? RDKit figures that out.
Okay, that's critical.
And it does something else really important. It extracts Bemis-Merkel scaffolds.
Scaffolds. Like the skeleton of the molecule.
Exactly. The core chemical framework. If you're hunting for novel drugs, you're often looking for new scaffolds, not just tiny tweaks on old ones. RDKit helps you find and categorize those.
Right. Searching for genuinely new structures, not just variations. Makes sense.
Yeah.
So you've got your potential drugs, their properties, their scaffolds. Then comes the simulation.
Then comes AutoDock Vina. That's the docking workhorse. It simulates how your potential drug molecule, the ligand, might bind to your protein target.
And it gives you a score, like how well it sticks.
Yeah, it predicts the binding pose and estimates the binding affinity. That score is sort of the central piece of the puzzle in virtual screening.
Okay. And this whole stack, PubChem, RDKit, Vina, so this is all accessible.
Pretty much. Anyone with a decent computer and some know-how can use these tools. That's the classical foundation.
But even with all that power, there's a catch, right? A limitation that classical computing runs into.
There is. And that's exactly why the quantum element becomes so important.
Okay, here's where it gets really interesting for me. To figure out the best way a molecule fits onto a protein, these programs like Vina explore countless possibilities, right? How the molecule twists and folds.
Right, using methods like Monte Carlo simulations, which rely heavily on randomness to sample different conformations.
But the randomness, it's not truly random. It comes from pseudo-random number generators, PRNGs.
Exactly. PRNGs are algorithms.
Yeah.
They're fast, efficient, but fundamentally deterministic. If you know the starting point, the seed.
You know the whole sequence. It's predictable.
Which means the simulation often gets stuck exploring the same easy paths, the lowest energy valleys in the landscape of possibilities. It creates blind spots.
Blind spots where potentially the perfect drug conformation could be hiding.
Precisely. Decades of searching might miss it, just because the pseudo-randomness never led the simulation there.
So how does this system fix that? By injecting real randomness?
That's the core idea. Using true entropy from quantum random number generators, QRNGs, these tap into actual physical quantum processes, things that are inherently unpredictable.
Like quantum tunneling or photon behavior.
Things like that, yeah. The randomness they generate is, well, fundamentally random. Unpredictable.
And where does this true random seed go? How does it change the simulation?
It's primarily injected into two key places. The conformer generation that's figuring out all the possible shapes the molecule can take, and then into the Monte Carlo sampling during the docking itself.
So instead of just exploring the easy shapes.
The quantum seed forces the simulation to consider rare, sometimes higher energy shapes, unusual twists, unexpected ways the molecule might fit. It's like instead of just walking the well-worn paths, you're suddenly bushwhacking into unexplored territory.
Okay, blazing new trails, I get that. But now the practical question. Quantum hardware. That sounds like something only huge labs or governments could afford. This system uses D-wave quantum annealers and IonQ ion traps. How does a solo researcher manage that?
That's a really important point. The system doesn't run the entire simulation on the quantum computer. That would be incredibly expensive and probably inefficient right now.
Ah, okay, so it's more targeted.
Very targeted. It's designed for efficiency. It calls the QRNG mainly for that initial seeding step. It uses the quantum hardware for what it's uniquely good at.
Which is what, in this case?
Generating that perfect randomness, for one, and also optimization problems. That's where D-wave comes in. Quantum annealers are specialized for finding the best solution among many possibilities, like finding the absolute lowest energy state in a docking simulation.
Okay, so D-wave helps optimize the search for the best fit.
Right. And IonQ, using ion traps, offers more general-purpose, gate-based quantum computing. While that's still evolving, it's useful maybe for more complex quantum chemistry calculations down the line, or even just for seeding now. The key is accessibility via the cloud.
So it's about using just enough quantum resource, just where it counts most to make a difference.
Exactly. Smart, minimal use of an expensive resource to overcome a critical classical limitation.
Okay, let's put the pieces together. What does the overall pipeline look like? The architecture?
It follows a pretty standard flow. Input, transform, filter, decide. But with that quantum twist.
So you start with selecting your target protein.
Right, target selection. Then protein preparation, getting the 3D structure ready. Then ligand selection, pulling candidates from PubChem.
And then the special step.
Then comes the entropy seeding, injecting that QRNG seed. That feeds into the docking simulation using Vina.
Vina runs the docking, exploring those wider possibilities thanks to the quantum seed.
Exactly. And the results, all the predicted poses and scores go into post-processing and filtering. It's very modular, very structured.
And this isn't just theory. The source material talks about a practical test run, right? Using a known cancer target.
Yeah, the 10-ligand EGFR docking run. EGFR is a really well-studied protein in cancer. The structure used was PDB-ID 1M17. This run is all about validation.
Making sure the system works as expected before you trust it with novel stuff.
Absolutely. You run a known drug that should bind well, like gefitinib. You expect a strong score, maybe negative 10.1 kmol.
Which means it binds tightly.
Right. And then you run something that shouldn't really inhibit EGFR, like aspirin, and you expect a much weaker score, maybe negative 7.4. If you get those expected results, you know your basic Vina setup is working correctly.
Then you run the real experiment.
Then you run your novel compounds, injecting the quantum seed during conformer generation and docking. And you compare those results, the poses, the scores, directly against a run using only the classical PRNG.
And you're looking for differences, not just a better score necessarily.
Not just a better score, though that's nice.
Yeah.
You're looking for how it achieves that score. Does the quantum seeding reveal completely new binding poses, new ways the molecule interacts, using potentially novel scaffolds that the PRNG run just never found?
Okay, I see. It's about expanding the quality of the search, not just the quantity.
Precisely.
But once you've done that, even with the quantum boost, you've generated potentially thousands, maybe millions of results, scores, poses, molecule shapes. That sounds like a massive data headache. How do you handle that overload?
Yeah, you hit the nail on the head. Even if compute time is cheap, analyzing millions of docking results manually, impossible.
So what's the solution?
AI. Artificial intelligence is the absolutely essential filter here. It's what makes sense of the flood.
How does it work? What kind of AI?
Different types. They might use natural language processing, NLP, trained on PubMed data, to assess the relevance of targets or compounds. They use graph neural networks, GNNs, which are great at analyzing molecular structures directly.
What do the GNNs do?
They're ranking the docking results, maybe filtering out molecules predicted to have toxicity risks, and critically, clustering the results, grouping them into those Bemis-Merco scaffold families we talked about.
Ah, so the AI helps identify not just good binders, but novel families of good binders.
Exactly. The AI's main job is compression and pattern recognition, taking this overwhelming output and boiling it down.
Boiling it down to what? What's the final output you actually use?
The goal is the wet list. That's the term used. It's the short, high-confidence list of compounds. You might start with thousands of docked ligands, but the AI helps you narrow that down to maybe the top 10 or 20.
The ones that look promising enough to actually synthesize and test in a real lab.
Precisely. The ones deemed worth the time, effort, and expense of wet lab work. Without that AI layer doing the filtering and clustering, the quantum entropy would just create an unusable data tsunami.
So it's really a trio working together. Persistence, the drive to do this, entropy, the quantum randomness opening up possibilities, and AI, the intelligence making sense of it all.
That's a great way to put it. Persistence plus entropy plus AI. That seems to be the innovation triangle here.
And what's really fascinating digging into the sources is that this whole engine, it's not just for drugs, is it?
No, and that's maybe one of the most profound points. The pipeline architecture. It's basically a general engine for structured exploration when you're facing uncertainty.
Meaning you can swap out the inputs.
Exactly. The core logic, inject randomness, simulate possibilities, filter results, doesn't fundamentally care if the input is a molecule binding to a protein or, well, something else entirely.
Okay, wait. Are you saying the same system, the same entropy injection principle used for finding drug candidates could be applied to something like forecasting the weather?
It sounds wild, but yes. That's the QuantumTornado case study mentioned.
QuantumTornado.
Seriously.
Seriously, it uses the same quantum seeding approach, but applies it to atmospheric data within weather models. Instead of looking for rare molecular shapes, it's looking for rare subtle collapse signatures in the atmospheric patterns. Signatures that classical deterministic weather models might smooth over or miss.
That's the idea. Those subtle patterns might be the earliest indicators of severe weather formation, like tornadoes.
And did it work? Is there proof?
The validation described is pretty staggering. In one instance, the QuantumTornado system apparently predicted the path of an actual tornado 4.5 hours earlier than the standard classical radar models.
4.5 hours, that's potentially life-saving.
It's a massive difference, and it demonstrates the principle beautifully. The core engine works for complex dynamic systems, whether they're molecular or meteorological.
That really broadens the scope. Are there other areas mentioned?
Yeah, material science is another big one. Think about designing new materials from the ground up. The Zabin-Carbone vision is referenced using similar principles to design things like advanced metal-organic frameworks, MOFs, maybe for capturing cohers efficiently.
Or finding better ways to store hydrogen.
Exactly. These are all fundamentally optimization problems, exploring vast possibility spaces where true randomness could unlock solutions classical methods might miss.
So the big takeaway here, for you listening, really seems to be the precedent this sets. This is one person, a retired engineer, essentially proving that the tools, the open source software, the cloud quantum access, the public data that are out there.
Right. It's not necessarily about waiting for some huge institutional grant or permission slip. It's about having the curiosity, the persistence.
And just deciding to build it, being the person who, as you quoted earlier, will not wait for permission to innovate.
And it's interesting, the architect himself, Ansari, is quoted as saying he felt sharper than I did at 30, even at 70. That personal drive, that persistence seems deeply connected to making this actually happen.
So we've walked through the classical tools, seen how quantum entropy provides a crucial edge by overcoming deterministic limits, and understood how AI is vital for making sense of the resulting data. It really paints a picture of a more democratized future for discovery.
It definitely suggests a viable path, integrating these different technologies in a smart way.
Which brings us to our final provocative thought for you. If one person, using tools accessible today, fueled by persistence and a bit of quantum randomness, can build a system that potentially finds novel drug scaffolds and forecasts devastating storms hours ahead of time, what breakthrough might you uncover?
What unsolved problem in your field, your area of interest, could benefit from that same combination of relentless curiosity, clever use of available tools, and maybe, just maybe, embracing a little bit of structured randomness?
The future discovery, it seems, is wide open for those who dare to build it without waiting for permission.